microphone-wise? Super, brilliant. I'll jump right in that case. Yes. So I'll be talking about some work that I did back now in 2021 for that Paleosynthesis Open Research Challenge that Cecily mentioned. And this was finding automated, reproducible ways of cleaning fossil occurrence data, which is packaged together in my fossilbrush art package. I originally wanted to call it Brush and Flossel, but that was vetoed by my co-authors, I'm sad to say. So right now I'm a postdoc at the University of Birmingham. I'm working on the diversification of ray-finned fish in the Paleozoic, so the most diverse living vertebrate group alive today. But I also have an interest in developing toolkits for computational paleobiology, which is what led to my association with Palaeoverse. And so in general, again, as Cecily sort of mentioned, my broad research interests lie with what life is living where and when, and how these patterns are changing, both through geological time and across geographic space. Because if we can get a handle on these basic sorts of patterns, we can start to ask more interesting questions like, why are these patterns are changing? What might be driving them? And so to get at these sorts of questions, we need to have data on where when our life is living. So the data focus of this talk today will be on fossil occurrence data. So just as a little sort of a snapshot of what that looks like, just so we're all on the same page, we're talking about data sets which record unique instances of the presence of an organism in time and space. Well, we also know what taxon that organism is. So when I refer to fossil occurrence data, you can be thinking about a data table which looks something like this. And so given the sorts of research questions that I'm then interested in, the workflows for a lot of my projects start off in a fairly similar way. I go to the paleobiology data full of beans and bright ideas and quickly find myself utterly despondent with the quality of the data that is available in there. It is usually some flavor or some combination thereof of incomplete, biased, or riddled with errors. And so I not only find my bright scientific interests stymied by data, I now find myself with the additional headache of having to clean that data to even begin to make it usable and explorable. And so just think then about some of the how these problems manifest. So I mentioned that with fossil occurrence data, we're normally thinking about how something is classified, what age it is, and where it's located in geographic space. So these sorts of the three axes we can keep thinking on throughout this talk: taxonomy, time, and space. And along each of these axes, we can be thinking about things like how incomplete our data it is, how it might be biased, and where our fundamental errors within that data might lie. So data incompleteness, that is a perennial paleobiological problem. It's never going to go away. We're never going to have the entire fossil record. All we can do is try and accumulate more data and better data. The other two factors, on the other hand, those are something where we can potentially be a little more structured, as it were, in how we go about addressing these issues. And because if depending on what kind of question we're trying to answer, we need to ask if our data is even fit for purpose in the first place. Because if we put poor data into analysis, we're going to get a rather meaningless answer back out again. So we're worried about the ways in which our data might be biased or wrong. I say biased and wrong. I actually think it's worth just quickly rewriting these terms and telling something which is maybe a nice little shift in perspective. I think in terms of, rather than saying biased and wrong, I will make this mistake myself in this talk. I think it's better to talk about things in terms of structure and anomaly, as these encapsulate a broader focus of what's going on with these data. So reterming it from bias to structure is actually something that Garwin Antel, a previous Palaeoverse Lecture Series speaker, inspired as a shift. And this is the idea that these biases in our data, they're not inherently negative things. In each of our taxonomic, spatial, and temporal elements, this data will have a structure which is a composite signal of the ecological realities that led to our data versus how it's been sampled. So sampling bias versus ecological signal, these are just inherent structures of the data. They're not necessarily positive or negative. It all depends what sort of questions we're asking. So I think a really good example, for instance, where we can And really a good example would be where we are interested in how the sampling bias itself is working. So papers, say, by Raja, looking at how colonial history has biased the sampling of fossil occurrences. That is something where we really care about sampling bias as a real interesting element of our data, and not some dirty word, as it were, for incomplete or useless. On the other hand, the anomalies in our data just throw our analyses into disarray. And so the reason why I want to use anomaly here is, again, to encompass data which is not necessarily just wrong, so say an incorrect taxonomic identification, where you also might have inconsistencies or things which are equally correct depending on the context in which you're speaking about it. So, for instance, spelling variations of names. Sure, there might be one which is a little more correct than another, but functionally we know what they're referring to. So inconsistencies and anomalies, I think, is a little more inclusive way to talk about things than just pure error. So in terms of these two facets of fossil occurrence data sets, structure versus anomaly, dealing and exploring their structure, dealing with and exploring their structure is it's a pretty well-known topic, and so correspondingly, there's quite a diverse landscape of tools out there for working with these data. Whether we want to divide our data up into different bins to start exploring its structure in terms of sampling or ecology, into standardizing our samples to make them more objective, more usable for comparative analyses, or whether we're trying to infer broader patterns from whether that is diversity dynamics with DivDin, sampling corrected diversity with iNext, or even taking things to more complex programs, again, like Pyrate. On the other hand, the ecosystem for dealing with anomalies in occurrence data, you could say, is perhaps a little less well developed. And in fact, where there has been development, this has been motivated pretty much entirely by problems and workflows by neontologists working on modern occurrence data, so from databases like GBIF, for instance. So a lot of the R packages you see out there for cleaning errors in occurrence data, they have been developed with modern neontological occurrences in mind, whether that is the biodiversity data cleaning package, TaxoDB, or coordinate cleaner. There is very little out there, basically nothing, for fossil occurrence data. And this is because fossil occurrence data presents its own unique challenges. So with fossil occurrence data, there are several things we need to think about in terms of if our data is correct and accurate before we go on to analyze it. And so the first of these is the dating of our fossil occurrences, their chronostratigraphy, just to give a sense of where problems can creep in here. So this chart on the right here is showing how time intervals, or two particular time intervals in the international chronostratigraphic chart have changed across what's that, 60 odd years of geological research. So essentially this is to make the point that chronostratigraphy can often be a set of moving goalposts. The absolute ages assigned to our intervals are under revision. Individual collections or formations have their ages revised time and time again. Granted, not necessarily to as an extreme degree as some of the revisions being made back in the 1960s, but these changes do still occur, and they can still introduce inconsistencies, anomalies, and errors into our fossil occurrence data sets. Similarly, when it comes to the taxonomic assignments in our data set, we cannot we can't really get genetic sequences from our taxa and see how different or similar they otherwise are. We have to rely on pure morphology, and when we're dealing with pure morphology, even though there is plenty of exceptional work out there to try and make morphological assessments of taxonomy more objective, more transparent, and more standardized, there is naturally still more scope for subjective disagreement for a litany of revisions, reassignments, and recombinations to creep in. Anyone who has cleaned a fossil occurrence data set will, I think, have a sense of this, is how frequently outdated or invalid taxa slip in there to complicate our analyses. But then the final point I want to make of this, and this is one of the key motivations behind fossilbrush, is how these two sets of problems can interact. So if we take the true stratigraphic range of a taxon, as it were, in green, all it takes, for instance, is a chronostratigraphic inconsistency or error to suddenly produce an anomalous range extension in the record of that taxon. But equally, if we misidentify a taxon in a much older time, then suddenly we have massively inflated the stratigraphic range of our taxon, all through error, inconsistency, and the interactions between these different things. But I am certainly not the first, nor will I be the last to think about these problems. They have been on the minds of paleobiology database users and indeed paleobiologists for a lot longer. And so this was a problem that was recognized by researchers from the Friedrich Alexander Universität Erlangen. And so the paleosynthesis team there put out an open research challenge in 2020, towards the end of 2020, just after COVID summer, when I had very little else to do, to try and produce a coding solution to the problem. So this was issued by the Saber Raja, Adam Kochis, and Wolfgang Kiesling. And the brief was to create an R package to clean fossil occurrence data in such a way that it is transformed from a manual, time-consuming, subjective, and really quite tedious effort into something which is a little less painful to deal with, shall we say. I'll stress now that fossilbrush is not a replacement. Nothing can be a replacement ultimately for true exhaustive checking of your data. Nothing can replace expert geological and taxonomic expertise, but we also have to acknowledge that when we're dealing with ever larger data sets, exhaustive cleaning just becomes infeasible. So we do need semi-automated and automated ways to start to tackle the data cleaning problems at the scales that we are starting to encounter with the Palaeoverse database. And so the solutions were published in 2022 in the fossilbrush R package. And again, you'll note here that this is referring to the automated detection and resolution of anomalies in paleogeological occurrence data, not error. The errors are certainly anomalies, but again, without an exhaustive cleaning effort, you cannot say definitively if it is an error or not. Rather, we want to focus on anomaly detection and flagging primarily, rather than true resolution. I'll just briefly also mention that on the kind of three axes I laid out earlier, geography, taxonomy, and time, one thing that fossilbrush doesn't do is check for geographic errors. And this is because actually the coordinate cleaner R package, even though it was developed for use on modern occurrence data, it actually can perform quite well for fossils too. It implements basic logical checks like are your latitude and longitude coordinates within the correct numeric range, and do those coordinates actually match onto the country, say, where a fossil occurrence is said to be from. Yeah, so it's a very nice general flexible tool, and I actually think it does have a couple of paleo-specific functions in there as well. So I'm leaving geography checking for now for the coordinate cleaner package. Instead, fossilbrush's remit is looking at taxonomy, time, and the interactions between these two facets of data, and in ways that can transform sort of anomaly detection from requiring exhaustive manual searching into something more akin to statistical anomaly detection. So just to jump into what functions the package offers, the kind of opening functions are very basic, very boring, but very important. They are just checking things are written and assigned correctly, essentially. So the check taxonomy function, again, this isn't saying your taxon is incorrectly classified or it's an outdated classification. That is for other tools, which frankly do need to also be developed in a more formal capacity for paleobiology workflows. Rather, it's just checking, do these names meet some basic properties that we expect of taxonomic occurrence data, and that is in terms of formatting and spelling. So for instance, for a species name, we would expect a capitalized genus name and a lowercase specific epithet separated by a space. But we can get all sorts of weird inconsistencies. You can get people chucking numbers as a placeholder species name. You can get an underscores in there. This is actually a double white space. That's a fun one, which can create complications for coding workflows. And these are all tiny, silly little errors, but which are very easy to resolve, not just with check taxonomy, even just with basic R functions, but they're things which are very easy to slip through the net unless they're checked for early and upfront. So this is what check taxonomy tries to do, is to automate some of this basic checking. What it also checks for similarities between names. So granted, one of these names will be correct, but equally, both would probably be used in the literature. And certainly in the paleobiology database, there are plenty of examples where there are spelling inconsistencies between things which should otherwise be the same taxonomic name. Already in this instance, we have gone from 1 unit of diversity to 2. So we want to make sure that these sorts of errors are ironed out as much as possible. We might also be interested in analysing diversity at higher taxonomic levels. So we will also check whether the higher taxonomic assignments in our data follow basic sort of logical expectations. So on the left here, this is one of these are outputs from fossilbrush actually, which I'll show you at the end of this if there's time. But we would expect our classification to remain discrete until they merge at some higher level. What we do not expect are for there to be multiple higher classifications of a lower taxon, but yet it does occur. Again, these are examples taken directly from the Palaeoverse database. So the check taxonomy function is trying to take care of all of these sorts of little initial formatting errors and inconsistencies in a reproducible, sensible way. The other thing we want to check is the dating in our data set. So this function is potentially going to be a little bit outdated, but at the time when I was developing it, the Palaeobiology database was still using stratigraphic ages taken from the 2012 version of the ICS chart. We're now a fair bit on from that initial dating point. When I was constructing this, GTS 2020 had just come out, so I thought it would be a good idea to provide a singular naming resources for all these stratigraphic intervals. But I want to take it a bit further. Rather than just provide support for names from the standard set of ICS intervals, I also want to provide support for all of the potential weird regional timescales, alternate dating schemes out there, things like the North American land mammal ages, for instance, or magnetostrat intervals, things which you do find in the Palaeobiology database in plenty of occurrence data sets in static published electronic supplements. We want to ideally have a singular resource where we can look up and assign consistent ages from. So Chronoscale currently supports, I think, about 1,200 or so interval names, including the ICS. So we take our data, we run it through these two functions, and hopefully we get something where the taxonomy and the chronostratigraphy, even if it's not free from errors, it's at least consistent between every occurrence in the data set. Once we've, just to show kind of the scale of this, this was when I ran these functions on the entire PBDB a few years ago. Most of these non-letter character flags, these are actually for the way the Palaeobiology database records no higher taxonomic levels, so they use underscores. So it's a little bit weird. It's not necessarily wrong, but it's things we can look out for in our analyses. For instance, if we were trying to do, say, tallies of diversity within the higher taxonomic unit, to be a little bit odd if we treated not a no assigned family as a taxonomic unit. For these other sorts of errors, so potential synonyms deriving from name spelling variations, there are a few things flagged there, and a few things again where the higher taxonomy, despite the PBD's very well developed infrastructure for recording taxonomy, we still get these little inconsistencies. So the PBDB is generally quite good, I think, in terms of really trying to keep data relatively consistent. I think where these functions really come into their own are where we are combining data sets from different sources, which may have used quite different standards. These are the cases where inconsistencies can very readily slip in, and we need a way to identify and iron them out. And so again, this is always the ethos of fossilbrush. These functions can provide automatic resolution of these of the anomalies they find, but the preferred usage of them is to flag data to the user to be manually checked. So again, it's not replacing expert manual checking, but it's guiding those efforts to where the errors are most likely to be. So once we've got our ducks in a row away, we can actually start to try and iron out some of these taxonomic and stratigraphic errors. One of the easiest ways to do this is to flag it against a set of reference ranges. This actually was, again, a sort of contrivance in the original FAU research challenge. They wanted data to be scanned and flagged against the Setkowski compendium as a reliable reference database. Whether the Setkowski compendium is still reliable is perhaps another question, but it's but you could use whatever set of reference ranges you have available, whether they're derived from the treatise of development paleontology, for instance, or from your own efforts and taxonomic expertise. And so this is implemented in the function flag ranges. This will take all your occurrences and taxa in the data set. It will match them against your set of reference ranges, and it will tell you whether that taxon or occurrence is within reference range or whether it falls outside of it. But it doesn't just tell us yes or no, is it correct or not. It tries to give us a little bit of information about just how incorrect these things are. So there are these additional error codes that are developed to try and inform you whether your occurrence partially overlaps or falls fully outside of that reference window. And these tell us something potentially, the magnitude and distribution of these inconsistencies, I think, can tell us potentially something quite interesting about why they arise. So if we look at these grey histograms here, the left-hand side are for errors for whole sets of taxa. On the on the left, we have whole sets of taxa, B here, whereas D are for individual occurrences. I think it's quite interesting to note that the error distribution for entire taxa is pretty symmetrical. There's nothing too interesting going on there. On the other hand, when we look at occurrences specifically, small errors on the first appearance datums, the upper ages of occurrences, seem to be much more prevalent than errors on the last occurrence. However, the last occurrence errors seem to span over a much wider range. And so I think within these signals is where we can find evidence for where things are, say, slightly poorly dated, or whether there is a genuine range increase relative to our reference set of ranges, or whether we have poor identifications and force to. I think this is the basis for this. So again, we want to flag as our default, but we can try and revise those ranges in light of that data. So the eagle-eyed amongst you will have noticed we do not just have grey histograms, but these red post-solve histograms as well. And so the way that this occurs is using this function here, revise ranges. What this will do is for a given collection of fossils, a PBDB collection or something specific to your data, it will identify all taxa in that all taxa in that collection with corresponding entries in your set of reference ranges, and it will then try and identify the broadest overlap between that set of reference ranges. If the number of taxa overlapping is above a certain threshold, the default is 75%, it will effectively use the ranges of the taxa in that collection as a calibration for what that collection age might actually plausibly be. And so this is then the result of revising PBDB collection ages based on this sort of calibration of Sepkoski compendium taxa. So we do get this high reduction in the frequency of anomalies, rather, not errors, relative to our reference ranges. But again, this is to say it's entirely dependent on whether you trust your set of reference ranges or whether you think there genuinely could be a fossil in your collection which represents a genuine range extension of your taxon. So again, this is stuff where you do want to be going back into some of the more anomalous collections and trying to manually confirm what these semi-automated flagging methods are telling us. So that's everything involving flagging against a reference set of ranges. That's a typo there, ignore that. The other methods try to use the internal structure of the data set in isolation without any reference to external data sets. And so the way it does this is based on what I am calling, or what I call in the paper, occurrence density distributions. And so this is the idea that if we look at sampling of occurrences through geological time and the biological history of the taxon to which they pertain, there should be kind of a broad biological expectation here. We should be expecting our taxon to originate in a discrete point in time. We should expect it to expand its geographic range and its abundance to some sort of peak, and then expect it to decline again through extirpation and eventually final extinction. And what we would expect the density through these sets of occurrence stratigraphic ranges to follow potentially is some kind of unimodal distribution, something like this. This is, I think, a not unreasonable idealized expectation of what the spatiotemporal profile of a taxon should look like. And so the densify function will take your set of fossil occurrences and it will calculate these density profiles for you. Once you have these density profiles, you can then run this second function here, find peaks, to identify where we expect the maximum, to find that numerical maximum. That numerical maximum will not necessarily be equivalent to its true maximum geographic range due to incomplete sampling. This is more just to identify a statistical property of this curve, which we can then use for our other detection functions. So this is our idealized expectation, but of course we are dealing with messy real world data. So to show you what that ends up looking like, we get something a little bit more like this. We still have ones which do conform to that expectation. So this plot here in the bottom left in yellow is for this Cretaceous turtle species, Cariamis. It meets that unimodal profile that we expect to see. This is largely because I think it's actually a singleton occurrence, but nonetheless, it conforms to our expectations. Where we then have splits and gaps in our peaks, these are the sampling artifacts we are then interested in using to determine how consistently sampled our taxa are through geological time, and then to use that consistency as a means of potentially detecting anomalies and errors in our data. So if I then move to the top right quadrant here for a very different taxon, Waptia, this stem new arthropod known for the Burgess Shale, we see in its occurrence distribution there are two peaks. So what's going on here? If you go to the occurrences in the PBDB for Waptia, you'll find there are actually two collections where this thing is sampled. One is the Burgess Shale at about 508 million years ago, but then the other is the Margam Shale in Utah. It's a little bit older. And potentially 1,000 miles further south. However, if you then go and look at papers which describe the anatomy and distribution of Waptia, they suggest that these two peaks, these are still the same taxon, and their idealized kind of unimodal range. This is an artifact of incomplete sampling. We just do not have the fossils of Waptia in this intervening time, but we would expect this to be a singular unimodal curve. On the other hand, if we now jump to this wonderfully charismatic gastropod, Nanokailina, this is the best photo I could find of it, I'm sorry. The two peaks in its occurrence distribution are separated by more than 150 million years, almost 150 million years. This may be where this is quite an extreme example, but where we might want to think of possibly these things are not the same taxon. This is where we have some kind of taxonomic error that may need to be revisited in our data. Perhaps these things should actually be very different organisms. What is the likelihood that this genus persisted unsampled for 150 million years? Potentially quite low. So these split peak methods give us a basic but quite effective way of quickly scanning really large data sets and just seeing how consistent are they, where might our errors lie. So I've saved the last of these distributions just because it's for quite a special taxon, as it were. So and it's for Lingula. And so Lingula, the reason why I want to really single it out, aside from its absolutely horrific occurrence density curve through geological time, this was also one of the organisms kind of exemplified as a thing to target by the initial paleosynthesis challenge. It is the poster child, the poster brachiopod for a dustbin taxon, where the lingulate flat elongate shell. This morphology has evolved so many times in brachiopod history for burrowing, and yet people have time and time again referred it to the same taxon, Lingula. So we surely do not have one single genus, Lingula, lasting from the Cambrian all the way to the present. Rather, what we're seeing here is multiple instances of the lingulate morphology evolving time and time again, but then being lumped together. So this is where we absolutely have taxonomic error, but where we can turn it from being a potentially subjective and manually exhaustive error identification exercise into something based a little more on anomaly detection, which we can then do statistically rather than manually. So once we have these potentially more complex occurrence density distributions, we want a way to start dealing with what they might represent. And so this is implemented in the threshold range function here. I mentioned that where the peaks in our density distribution lie, these are quite an important element of these structures. We can use these as a rule of thumb to say whether an occurrence density curve is being split by incomplete sampling, or whether two peaks actually represent lumping of what should be separate taxa. And so what the threshold ranges function will enable you to do is to scan forwards in time through an occurrence density curve. And each time it finds a peak, it will then search for the distance to the next available peak. If that interpeak distance, as I'm calling it, interpeak threshold, if that distance falls below a certain threshold, it will treat those peaks as effectively the same taxon, but potentially split by incomplete sampling. Where the distance between these peaks exceeds that threshold, that's where it will suggest that these actually could belong to different taxa. And so again, this is absolutely not replacing proper hard taxonomic effort. It's merely trying to guide the user to see where there may be inconsistencies and outdated elements of their data set, which they can go back and investigate. The end result of it, though, is this sort of series of split curves, these thresholded curves. You could consider them chronospecies or just things with similar morphologies which are split out of the wastebasket, however you want to view it. But then you can try, and then you can reanalyze these as true pseudotaxa if you so wish, or again, just simply go back, take a look at the data, and try to understand where these errors and inconsistencies might be coming from. And so then the final method I want to draw attention to is the final method available in fossilbrush. This takes density curves again, occurrence density curves, but it tries to do something slightly different with them. And so the inspiration for this method actually comes from something a little bit older and from the world of microfossils. So in 2012, David Lazarus and colleagues published this cool little paper describing a method called Pac-Man. And so Pac-Man was developed to identify anomalies and errors, familiar language, I hope, in microfossil assemblages taken from drilling cores and things like this. And so the logic behind Pac-Macro is that as you measure the abundance of individual point-wise occurrences in a sediment core, you can construct again these density distributions of them. But what we expect due to taxonomic misidentification, due to difficulties in delineating morphological boundaries between descendant and antecedent species, and potentially to reworking of sediment, the errors in these sediment cores we expect to be concentrated towards the tops and the bottoms of the species distributions. And so their solution to this, hence why it's called Pac-Man, was to trim from the top and from the bottom of the core using this little Pac-Man sort of face here. And the way they implemented it was to do just a straight sort of thresholding. We will remove the top 2.5 and bottom 2.5% of that occurrence distribution as suspect. So again, I think this is why it's quite a useful exercise to think that this isn't truly identifying fixing errors. Rather, this is excluding entirely what may be unreliable data at the cost of potentially slightly underestimating your taxonomic range. Nonetheless, I think because it is a statistically implementable procedure, I wondered, could this be adapted for macrofossil data? And so again, to relate this back to the density curves I mentioned previously, this is the basis for what I called Pac-Macro. So Pac-Man applied to macrofossils or macro ecological data, as it were. And obviously, with a sediment core, you're talking about point-wise occurrences where you could put, the tip of the pen or a needle exactly where that fossil lies in the depth of the core. Fossil occurrences, or macrofossil occurrences, will generally be a bit different. They will usually have some sort of age uncertainty associated with them. So the way we get around this, the way we kind of make an equivalence with the original Pac-Man method, this is actually the basis for how you construct the density curve in the first place, is you would take your taxon range, you would sample along its duration at very fine intervals, like at potentially 0.1 million years, and you would use those kind of samplings as point-wise pseudo occurrences for which you construct your curve. From here, we can now actually we can then start to employ the original logic behind Pac-Man, and that's to say that we expect errors in our data to be more likely to occur towards the tops and the bottoms of the stratigraphic range of a taxon, where things have been misidentified and you have suddenly introduced a 10 million year range extension due to taxonomic error or imprecise dating of the fossil collection, for instance. I don't think, because these are ultimately ranges associated with occurrences rather than point-wise observations, it's not necessarily the most justified thing to do to simply remove the top bottom and the top 2.5% of the curve. So I want something a little more principled for trimming macrofossil occurrence data. And so the way that PacMacro does this is it will take your density curve. It will identify the 95% distribution of that curve. You can modify that value in the function to be more or less stringent. And what it will then do is it will measure the temporal length of your tails relative to the duration of your 95%. And above a certain percentage, I think the default is 30%, it will say 30%, your tail is 30% or more of the 95% duration of your taxon. This is potentially something for you to take a look at and see what's going on. So this example here is for the coral lithostrotion. And so the true well-established temporal range of lithostrotion is, I believe, early to middle Carboniferous. That is where we find peak lithostrotion diversity. We do not find lithostrotion out of that sort of range. Even the family Lithostrationidae, that I think is almost overwhelmingly a fully Carboniferous family. Lewis might be able to correct me on that later. Corals are not immediately in my bag. But then we have this very clear anomaly located in the last appearance date of tail. And when you go into the paleobiology database, this may have been fixed since. I say this is from the paper rather than from the most recent data available. This is one occurrence, I think, of lithostrotion from Indonesia with an assigned temporal uncertainty of Permian to Triassic. So who entered that into the database, I do not know. It's a rather poorly resolved data point, I think, to include in the first place. And if you were doing a download from the PBDB, chances are you would already be filtering by some kind of maximum upper threshold for how uncertain you can tolerate your data to be. But equally, this is quite an extreme example. You can imagine how you may still have anomalous tails occurring at much smaller temporal scales due to slight dating errors in collections or taxonomic assignments just above or just below the true temporal range taxon. So I think I've gone through that fairly quickly. I think that then gives plenty of time just to quickly demo some of the functions in the package, just so you have a sense of what the outputs look like. So I'm just going to I'll stop sharing very briefly, and I'll reshare our studio just to then, give you a little showcase of these functions. So I'm going to now try and share studio. There we go. Could someone just give me a quick thumbs up on the Zoom? Oh, thanks, Will. Brilliant. Super. There we go. All right. So just a little sort of script here. These are basically all taken from the package examples. So what you see is what you get. There is also a vignette available through fossilbrush if you want to have some slightly more detailed case uses and some more of the caveats available what fossilbrush does. I mean, if you are really interested, do also go and read the paper. Always nice to have reads and citations of the work. But then to take you through the workflow, so we have our studio script. We have our script here in our studio. We load the fossilbrush package in the way we would expect. Fossilbrush comes with an inbuilt data set. This was called Brachios. This is just a straight rip of brachiopod fossil occurrences, all the brachiopod fossil occurrences in the Palaeoverse database when I was developing fossilbrush. So you can see it's just 150,000 odd brachiopods from all across the Phanerozoic. I mentioned earlier that the Palaeoverse database is actually not too bad in some regards at trying to make data, if not correct, but at least consistently formatted. But there are still errors to be found even in this static YAC, even even before we actually get into, I don't know, is Darbia still a real taxon? Has it been synonymized to something else? Of course, that is where you could go to the paleobiology database itself and query its taxonomic opinions therein. But again, but as I say, we're not interested necessarily in whether something is truly correct. We're identified in detecting whether it is anomalous or not. Just to show the first function here, check taxonomy. You would supply it with your input data set. Minimally, this just requires there to be taxonomy columns, so the very ones which are just displayed here. So we tell the function our data set contains the following taxonomic levels, phylum through to genus. It will by default, it should by default, I think it's set to use the genus as the focal rank of which to scan. I think you can set it to other things. I mentioned that this function will do a few different things. It will check whether our taxa are consistently classified at high taxonomic levels. It will scan for weird little formatting errors and things like that. But it will also scan for inconsistent spellings and formattings and taxonomic names. And so it does this using kind of text processing tools and fuzzy string methods. These aren't things I've developed. These are implemented in a bunch of our packages, and they're quite standard tools in broader data science and text editing, but they're very useful, statistically supported ways of identifying just how similar words are. Of course, one problem we have potentially with fossil occurrence data is pretty consistent reuse of common Latinate or Greek prefixes and suffixes. So to avoid the function being thrown off, we can also tell it at a given taxonomic level whether there are particular commonly used prefixes and suffixes to exclude miscalculations. So this just helps tailor the use of the function to the particular data that you're using. So if we just quickly construct these lists, so we have our taxonomic ranks, we have our list of suffixes to aid with synonym detection. We can now run our function. So it'll run this 4-step cleaning process. It will check for our formatting errors. It will check for our spelling variations. It will check to see whether taxonomic names are discrete to their supposed rank. So we don't have a genus name being reused at family level, say, something which could very easily slip into a hand-compiled data set, for example. And finally, it will check if our taxonomic classifications are consistent throughout the data set. So just to show you the output of this, which is the check. There we go. That is just a list of 4 things containing each of our sets of checks. So if we have a look at ranks, nothing really to be found in here. That's great. So there's nothing inconsistently classified at different ranks. If we have a look at our formatting errors, what this actually returns are the row numbers where there is something problematic and the column in which it occurs. So if I just index one of these very quickly to show you what's going on. So if we do brachios, let's have a look at what's going on in the class. So that's the column we want to look at. And so we now need to get the rows of the data set which are potentially problematic. So we can just use the index which functions return to us. We check by, what was it? Formatting. Let's have a look at non-letter ones and class. And so this is the example as I mentioned earlier, where actually this is not an error per se, but it is something where a higher taxonomic name, as it were, is formatted in a way which does not conform to our typical expectations. So again, we would not want to use no class specified, for instance, as an analytical unit if we were looking at high level diversity. Just to breeze through the last couple of these because I don't think there's much to see within them. And this one is easy, yes. So we also have a plotting function to visualize the taxonomic classifications of things. So if I just quickly plot this one here for the brachiopod species Atropa, this gives us a classification chart which conforms to our expectations. Everything is just flowing upwards in a nice discrete manner. However, we can have a look in the output of check taxonomy for where things are inconsistently classified. So again, bearing in mind, this is data which is in the paleobiology database. And so even though it has this generally quite good system for ensuring that things are incorrectly classified, if we now have a look at what this error shows, there we go. I think my Zoom control should be out of the way. Brilliant. There we go. So you can see that this is an instance where there are conflicting classifications in our data set that we want to iron out prior to higher analysis. And just to show you the final one, because I think it's quite a nice example of, oh no, let me check. By synonyms. So this is our synonym table here. So what this tells us is the taxonomic level at which that synonym occurs. It gives us our two synonymized potential synonym names in columns T1 and T2. And we also have a couple of additional columns. So we have the frequency at which each of those names is used, and then a numeric measure of actually how mathematically similar it is. You can change which numeric method is used for this, so it's quite a good thing to compare between methods. But just to show then an example of how you might go about using this, we can say, take a look at lingula, lingula versus lingularia. These are quite similar these are quite similar names mathematically, but they are distinct taxa. On the other hand, if we look at the very last thing printed here, Sphenospira versus Sphenospira. And if you go to the literature, these things are indeed the same taxon, but this is where we have an inconsistent spelling, which our check taxonomy function can quickly identify and flag to us. So I would say cleaning is a boring thing to do, but this hopefully makes it quicker and less painful to go and track those errors down in a manual manner. Nothing much to say with Chronoscale other than that it gives you the dates you want. So if I'll just run this as is to show what the output is. So you'll give it your data set. You'll tell it which columns you're trying to match names against, and it will return to you it will return to you those ages as new fad and new lad. The chronostratigraphy table in fossilbrush is something I'd actually like to redevelop at a later stage to provide more dynamic support for more recent versions of the timescale. But at the very least, it is quite useful for covering the whole host of weird and wonderful stratigraphic intervals, which aren't necessarily in other packages. Those are just appended to the end of the data set by default, as we saw. What it will also do is it will tell you where there are interval names which have not been matched in the lookup table. So that you'll just get NAs for those. You'll get NAs for those numeric entries. But it will at least tell you where those are. So again, you can go back, check, and work out what might be going on there. Why is your name failing to match? Is it a spelling error? Is it inconsistency? Is it because the name is just not present? So that's a way of going about tracking that down. So again, just to quickly breeze through the other checking functions that we have. So we'll load the Sepkoski compendium as our set of authoritative reference ranges. Believe that or not as you like. We'll trim that to the brachiopod portion of the data set. And now we'll run the flag ranges function. So this runs fairly snappily. So 2,200 taxa in our current data set to check, or at least 2,200 taxa odd, which had matches in our reference data set. And then we'll have a look at what that output looks like. So first off, it will give you the error status for overall taxa, whether entire taxa fall within your reference range, above it, below it, or overlapping, depending on those reference codes that those error codes that I showed you on the slide. And then you will also get the same output for your individual occurrences and just how different those ranges are. So this is again something where you could say, show me all occurrences which are discrepant by more than 20 million years, and that already gives you a quicker way of scanning and finding the more erroneous problematic bits of your data set. Anomalous, should I say, not erroneous. I said I would make the mistake. And then just to show what the run looks like, you can then take your reference data set and your error-coded data set, and you can try and use that to revise those ranges as a calibration of your reference ranges. This one will take a little while to run, so oh no, it'll be fairly quick. Oh no, there we go. This one, yeah, this is one which does take a little while to run. So I won't run that for the entire thing. But to say what you'll get out of it is your same input occurrence data set, but with a new set of age columns, which have split up your occurrence density distributions into their sort of discrete groups, as well as the error codes associated with those occurrences. Moving quickly on to the density methods, just in the final minute or so of the talk. So the densify function that constructs our density curves for all of our taxa. Once that's run, you can then use the find peaks function to numerically detect the peaks in that thing. It does that using both histograms and what's the other one? Histograms and smooth density curves, so you can use both methods. And if I just and so then we can then apply the interpeak thresholding method. So where we scan forwards through our occurrence data set, our occurrence density distributions rather, using each successive peak to evaluate, is this an occurrence distribution that is likely split by sampling, or is it potentially something where we have lumping of what are actually different taxa or taxonomy misidentifications. You can plot your density curves with this plot D profile function here. So if I just run this very quickly, and this gives us quite a nice example of where we have a split peak. Do we necessarily believe that Davidsonia lasts 50 million years? Maybe, maybe not. But again, this is where you would go to the literature and check if you think this, if this is raising alarm bells for you. And so it's very finely. Oh, and you can add the detected peak on here with the ITP function just to see which peaks it's detecting as relevant as the relevance to scan through. Finally, pack macro ranges. Again, quite a simple output from this one. It will tick forwards through your data set. Again, bearing in mind, this is going through 150,000 occurrences in a few seconds, so it definitely can scale well to large data sets and to iteration over different function parameters if you want to test sensitivity for that. And if we look at what we find in PACAM, what we get from this is the stratigraphic range in our occurrence data in the first two columns, the stratigraphic range represented by the 95% distribution. And then it tells us whether the tails in our distribution were marked as anomalous using, I think, a 30% tail length and a 35%. So again, this is customizable if you wanted to use a more or less stringent value. So I've rattled through the code quite quickly. That's all from me on the code and the PowerPoint. So I will stop sharing my screen, but I absolutely welcome any questions on this. I'm very happy to dive back into the PowerPoint if there's anything you'd like to talk about there too. And to say thank you very much for listening. It's a real pleasure to present fossilbrush after quite a few years of not actually paying it much attention beyond using it in the old workflow. And yes, do welcome any questions. Thank you.